we be in 50 years? What will we have to worry about? 500 years ago, we might not have been as lucky as to live past, say, 30. And you know what? The methods for even dealing with the disease back then were not quite pleasant, might I say. Like bloodletting, which is just not very nice. We don't really have to worry about dying from diseases anymore, such as polio or influenza. And even for someone with a deadly disease, we have methods to extend their life expectancy. Medicine is always making giant leaps, and in the future, we may not even need to worry about dying whatsoever. Welcome to Module 1.4 of my biotechnology course. I'm your professor, Dr. Thompson, and today we'll be looking into biotechnology used in clinical medicine. I hope you all did the reading, as there will be a test tomorrow, 50 multiple choice, 50-word answer, 50 paragraph answers, of course. Firstly, I'd like to ask, Mr. Weeb, to you over in the corner, what are the targets of biotechnology?
DNA, RNA, and protein.
Looks like someone's been doing their reading. For those of you who haven't done the reading or, haven't even bothered, some kids, today's lecture should enlighten you. There it is. Oh, how unfortunate.
Come on, Professor.
Technology is not my strong suit. Yeah. Biotechnology used in healthcare today is a promising area of science, utilizing protein, DNA, and RNA, said as Mr. Weeb so kindly, and as well as protein transcription and translation for diagnostic or treatment-related therapies. These products are developed by a host of genetic engineering techniques and methodologies specifically directed towards the physiological modification of cellular end products and/or processes. Although biotechnology is providing extremely powerful tools capable of altering the very basis of life for public good, of course, numerous ethical questions of wielding such power are also being raised. Biotechnology techniques being used in clinical therapies are enhancing the results of current treatments and have the potential to alter the pathway other risky treatments take. Biotechnology poses a significant primary treatment against genetic disorders such as cystic fibrosis, infectious viruses, or types of cancer by either inhibiting the expression of dysfunctional genes, tackling the processes certain viruses take, or by preventing cellular mutations and mistakes made in cell replication. On a more pathogenic-based lens, biotechnology can be used to harness the already existing characteristics and processes of bacteria and viruses, and even mimicking them in order to understand and utilize their pre-existing characteristics. An example of this can be found in the well-known CRISPR. This blob of an enzyme came from the all-feared bacteria. When bacteria are attacked by a bacteriophage, they capture snippets of the DNA and put them into something called a CRISPR array. This is so that the next time the bacteria is attacked, it can produce an RNA version of that viral DNA. They then use Cas9 to disassemble the invading DNA. This just goes to show that an understanding of genes through biotechnological and therapeutic means is the key to treating and eliminating diseases, whether they be viral, genetic, or bacterial. The purpose of this module is to A, provide insight into current and future biotechnology, and B, how it serves to assist in therapeutic treatments against inborn or pathogenic-based diseases and processes. Unlocking the full potential of our genes is the key to ending generations of genetic disorders and correcting mutations and mistakes made by ourselves. Genes hold the data for all living organisms, I hope you've heard, and contain the coding used for the certain creation of proteins, enzymes, and other secretions and products, all necessary for the survival of the species. To understand the importance of these genetic codes, scientists need to know the damage they can cause when mistakes are made to properly understand how we can reverse it, or better yet, prevent it. Genetic disorder is a genetic mistake caused by one or more abnormalities in the human genome. There are multiple types of genetic disorders, such as mutations caused in one gene, which is a monogenic disorder, mutations in multiple genes, multifactorial inheritance disorder, mutations in the mitochondrial DNA inherited from your mother, which is maternal inheritance, or by damage to the structure of entire chromosomes. The genetic disorder can be caused by environmental impacts such as UV radiation, or by inheritable factors such as recessive gene expression and mutations in genes, or by genomic imprinting. These mutations caused by mitotic and environmental events can lead to permanent damage to genomic data. And you know what? Even though they look like pieces of knotted spaghetti, genes are really important because they'll hold the instruction for the key of making proteins and enzymes for both unicellular and multicellular organisms. Just the slightest change in the DNA of an organism could have massive effects, like having a switch that once flipped causes a genetic disorder to occur. A monogenic disorder is the result of a single mutated gene. There are over 6,000 human diseases caused by the mutation in a single gene. These disorders can be passed down through generations and have been shown to influence inherited patterns. Such monogenic disorders include cystic fibrosis, sickle cell anemia, Fragile X syndrome, Duchenne muscular dystrophy, and Huntington's disease. Cystic fibrosis is an inborn disease that primarily causes damage to the lungs, but also the kidneys, liver, and the majority of organs in the digestive system. Cystic fibrosis affects cells that produce mucus, sweat, and digestive acids, and products required to digest food properly. In order for a person to develop the disorder, they must have 2 copies of the defective gene, one from each parent. If they were only to have one, they'd simply be a carrier, meaning they couldn't actually have the disease, but they could still pass it off to their children, which isn't very nice. But. The disorder is caused by the expression of mutations in both parent copies of the CFTR, cystic fibrosis transmembrane conductance regulator gene. A healthy CFTR gene provides a CFTR protein for organ membrane cells that create mucus, saliva, sweat, and other important digestive enzymes for organs in the body. For patients with cystic fibrosis, secretions that are meant to be thin become thick. This lifelong disorder can be tested by a sweat test, which measures the sweat viscosity of the individual. The most common mutation that leads to cystic fibrosis is the deletion of the 580th base in the CFTR gene, as shown in the image, which leads to the occurrence of a frame shift mutation. So frame shift mutation causes bases in a gene to shift, affecting how they'll later be read, which could have massive effects. Gene therapy has a number of techniques, and one of the most promising is CRISPR, Cas9 prime. Both of these methodologies, gene therapy and CRISPR, are related in various ways, but vary in their efficacy and accuracy in what you use them for. Biotechnology approaches cystic fibrosis by means of gene therapy. Gene therapy is a genetic editing therapy that relies on a viral vector to insert new genetic information into the patient's cells. The healthy DNA sequence will be placed into the infected gene beside the mutated DNA with the aim to express the healthy sequence instead and start to create the proper CFTR proteins again. The production of these proteins will allow the endothelial cells to manage mucus and prevent a buildup of thick fluid. Gene therapy is an experimental technique used by scientists to correct defective or mutated genes responsible for the development of disease. It is used to add genes, replace genes, or turn on specific genes that can cause cancer, different genetic disorders, or genetic diseases. Some gene therapy trials have used viruses to correct genes by adding unmutated versions of the same gene to the patient. Gene therapy has had major success in laboratory experiments and has the potential to provide future cures for monogenic disorders such as cystic fibrosis, or even Duchenne muscular dystrophy, Huntington's disease, and other disorders such as hemophilia A, severe combined immunodeficiency, and possibly even types of cancer. However, there still are a few speed bumps to overcome with the use of viral vectors. The body's natural immune response sometimes impedes the virus from reaching its target cell and sometimes causes complications for the virus. Some viruses, such as gamma retroviruses, add genes at seemingly random locations in the host chromosomes that can disturb other cellular functions and may lead to cancer. Adeno-associated virus-based vectors, big name, but they're also much safer because they always deploy genes in the same area of the chromosome, with exceptions in various diseases such as Alzheimer's. Gene therapy relies on viral vectors to vary the genomic data to the specified spot on the genome. And scientists do this by using 4 main viruses in gene therapy: adenoviruses, adeno-associated based viruses, retroviruses, and lentiviruses. Scientists are also studying the use of stem cells and liposomes in gene therapy. Stem cells could learn to fight certain diseases in a lab, and liposomes could be used as a certain different form of vector to vary the new genetic information. Gene therapy could possibly strengthen the capabilities of the immune system by correcting genes that are responsible for the failure of development of white blood cell manufacturing regions, which in turn are responsible for our innate and adaptive immune systems. CRISPR-Cas9 prime is the most powerful tool in terms of genetic editing to date, and also poses as a great tool for correcting gene disorders, and possibly even cancer, by accurately replacing, altering, or adding DNA sequences. There are 3 tools used by CRISPR-Cas9. They include a guide RNA, reverse transcriptase enzyme, and a Cas9 nuclease. The guide RNA is used to guide new DNA, hence the name, to its selected location in the genome. The guide RNA also contains new nucleotides to replace a faulty DNA sequence. A hobbled Cas9 enzyme cuts one end of the targeted DNA sequence, but not both, hence why it's called a hobbled DNA gene, to ensure the DNA machinery notices a change. The next enzyme, which is fused to the Cas9, reverse transcriptase, takes the new RNA nucleotides carried by the guide RNA and creates a copy of DNA, which replaces the old nucleotides at the site. If you can read that, that's pretty impressive. I need my glasses for that. Because one end of the new DNA sequence was left undone, the cell's machinery comes into play and either mends the 2 broken ends or takes a random nucleotide floating around the cytoplasm and places it as a sort of connector piece, which is kind of lazy. But anyways, another option is for scientists to add another nucleotide segment for the cell's machinery to grab onto and put into place for the scientists. Because of a small mismatch in the DNA sequences, the prime editor nicks the end of the sequence, which prompts the cell machinery to finish the sequence based off of the new DNA strand. Infectious pathogens such as human immunodeficiency virus, aka HIV, remain problematic for our immune system and our immune cells. HIV is a retrovirus that specifically attacks and degrades the human immune system, allowing other small diseases to easily overwhelm the immune system, finally leading to acquired immunodeficiency syndrome, aka AIDS. Current pharmaceutical treatments use highly active antiretroviral therapy, known more commonly as HART. HART is an antiretroviral drug cocktail that helps to inhibit the functioning of retroviral enzymes that are critical to the virus's reproduction. This treatment allows the human immune system to repair the damage done by the virus and prevent further infection. Now, biotechnology approaches this with monoclonal antibodies, as I'm sure you've heard of sometime. The specific monoclonal antibody 3-DNC117 inhibits the HIV from infecting the CD4 helper T cells by binding to a piece of the HIV spike proteins. So you see up there those green mushroom-looking guys? Those are what the antibodies are going for, and they're also what attach to the CD4 cell. It's very effective for HIV-positive patients in combination with HART and monoclonal antibodies together, and this could increase the efficacy rate of treatment of HIV together. Okay, I hope I haven't lost anyone. I swore my last class pulled the fire alarm on purpose. As you may recall from our last module, when we discussed the immune response, we talked about antibodies and antigens. Antibodies are tiny molecular Y-shaped proteins that are produced by the immune system to search for specific antigens, very specific. They attach to these antigens and then broadcast its location to more specialized cells that either engulf it or bombard them with molecular chemicals, which is pretty cool. Antigens are foreign viruses, toxins, or substances that cause the immune system to manufacture these antibodies in the first place. Monoclonal antibodies have 2 main parts. The orange variable region on the antibody is what recognizes a specific piece of the spike protein of HIV, in this case the green mushrooms, and the blue constant region is what is visible to the immune system and is what triggers an immune response. These tiny proteins are created in a laboratory by hybridoma cells. Hybridoma cells are created by fusing immortal melanoma cells and natural antibody-producing B cells. Hybridomas create mass amounts of a specific type of monoclonal antibody for therapeutic research and use. Monoclonal antibody binding targets are all identical in that they all target a very specific piece of a very specific antigen, unlike polyclonal antibodies. Millions are created by a single cell to bind to viral antigens, thereby inhibiting them from spreading and infecting. These antibodies form a clump of viruses, quite a mess, and they can easily be identified and then engulfed or destroyed by immune cells, such as macrophages. It's like a very messy puzzle that then gets destroyed. It's very messy. Monoclonal antibodies are being utilized in many current treatments and therapies targeting cancer, rheumatoid arthritis, cardiovascular disease, autoimmune disease, multiple sclerosis, viral spike proteins, and many more diseases because of their proficiency and accuracy in targeting and broadcasting very specific antigens. Monoclonal antibodies used as a treatment for infectious pathogens cannot be fully supported due to their efficacy rate dropping over long-term treatment periods. And this is because the immune system is starting to look at them and they're like, who are these guys? Like, these are kind of weird. What are they doing? So they just start attacking them. That's why heart and monoclonal antibodies, they want to use them at the same time to reduce the resistance of the immune response. Cancer is one of the most difficult diseases to treat. Normal cells develop mutations in genes, often involved with the regulation of the normal cell cycle to become precancerous. These activated precancerous cells undergo further mutations and evolve to dodge the attempts of our immune system to remove these errant cells. What we know as cancer is the summation of all of these mutations in the genomes of our cells that have led to the process of tumorigenesis. Further mutations can enable these tumors to metastasize and invade other tissues and organs. Cancerous cells inhibit the function and growth of natural cells and interfere with natural body functions. Cell mutations can occur because of mistakes made in the synthesis of DNA or from environmental impacts such as UV radiation. Cell mutations can lead to cancers such as leukemia and lymphomas that can sometimes be passed on through your genes. Cancer can be incredibly difficult to treat because different types of cancer respond differently to treatments, as they're composed of different people's mutated DNA. And cancers are notorious for learning how to evade certain drugs. A promising new approach to blood cancers is CAR T-cell therapy. CAR T-cell therapy is a clinical therapy that involves removing the patient's cytotoxic CD8 T cells, the assassins of the immune system, also known as natural killer cells. And what they do is they insert new genes into these T cells for a new receptor called a chimeric antigen receptor, or a CAR. These newly enhanced T cells are cultured and reinserted back into the patient and can now target, attack, and kill specific cancer cells. The therapy adds an additional receptor to the cell surface that specifically deals with lymphoma or leukemia cancer types. And while this therapy has potential, it currently remains very expensive, unsafe, and risky. But a benefit to the therapy is that the CARs, chimeric antigen receptors, cause a permanent T cell adaptation, which may come in handy if the cancer somehow comes back. As research into genetics furthers, scientists have recognized that not all carriers of genetic disorders are bad. And this is when it gets interesting, if you're not already interested. For instance, sickle cell anemia, which is a recessive disorder. If both parents were to pass on the genetic mutation, it would result in the genetic disorder occurring. But if only one parent passed on the mutation, the result would lead to the resistance of the malaria parasite. This suggests scientists take a careful approach to genome editing tools such as CRISPR and gene therapy because of the possibility of eliminating a potentially useful adaptation or mutation. Antibiotics. Okay. You see those pills in your house? Maybe your doctor prescribed them, or like your dog ate them off the carpet, which is like not ideal. Antibiotics are a natural or human-made drug capable of destroying or interrupting bacterial reproduction, meaning against life. They are currently seen as the most effective agent used against infection. They may seem like little pills, but they certainly pack a punch against bacteria. However, though, of course there's always a however, they have become less and less effective. Anytime an antibiotic is used, it risks the chance of the bacteria gaining some form of resistance against the antibiotic. As antibiotic resistance is becoming more and more prominent, we are running out of types of antibiotics to fight bacteria, and then our major treatment will become no longer useful. Kind of sad. These guys are the absolute worst, and I don't think many of you have heard of them. They're like super soldiers that evolved to specifically give you a very hard time. Superbugs are a strain of bacteria that are resistant to human-made drugs and antibiotics. They can become immune to multiple types of antibiotics and can even mix with other species of bacteria, which is just way more fun, through something called horizontal gene transfer, which I'll explain later, to become even more dangerous and incurable, or possibly more contagious and spreadable, which is lovely. The reason why superbugs are so dangerous is because even the most simple bacterial infections can become incurable, and then they can be fatal. Okay, Mycobacterium tuberculosis, depicted here, these little like long, elongated jelly beans, are the world's most deadly superbugs today. This specific bacteria alone contains a waxy self-surface made of something called mycolic acid. This bacteria alone causes 1.7 million deaths every year, as it has evolved to evade antibiotics and drug-related therapies. The main reason why it's so dangerous is it hides in the host cells like a stealthy assassin, which is kind of unfair, until it decides that there are enough others of itself to attack, which I'll talk about later. Horizontal gene transfer, also known as lateral gene transfer, is a natural phenomenon used to transfer genes between multicellular or unicellular organisms, instead of genetic information being transferred from parent to offspring, hence vertical gene transfer. Genes get transferred directly from one organism to another, like me giving Pam my height. This is an important factor in the evolution of organisms. HGT is also a major factor that leads to antibiotic resistance among bacteria, and it also leads to bacteria becoming more virulent. HGT is also a major factor that leads to antibiotic resistance among bacteria, as I just said. Bacteria containing certain genes responsible for antibiotic resistance can transfer their genes to other species of bacteria through mechanisms called transformation, transduction, and conjugation. In transformation, DNA floating around in the environment is picked up and fused with the bacteria's own DNA, which is like so sloppy and weird, but transduction occurs when a bacteriophage, or a phage, accidentally inserts the resistant gene for an antibiotic or drug directly into the bacteria. And conjugation is when cells exchange DNA via sexual pillows. The processes can arm yet another bacteria with immunity against that certain compound. And even though this may seem harmless, bacteria reproduce at an exponential rate and can be passed on to different people quite easily. Considering what I just said, if conditions are favorable, bacteria such as E. coli can reproduce every 20 minutes, which translates to one bacteria producing 8 others in an hour, which translate to 2,097,152 more E. coli in just 7 hours. And in 24 hours, it's 2 to the power of 72, but that broke my calculator, so can't show you that. Which is like a lot. I can't even count that high. That means if a single bacteria's genome mutates to become resistant, in like no time, it'll just be like an army of annoying little midgets. Very annoying. Because of this mass reproduction rate, scientists are struggling to slow the spread of antibiotic resistance, because not only do bacteria spread their resistant genes to their offspring, but also to other species of bacteria. Because bacteria can genetically alter themselves, they can sometimes become more dangerous branch of bacteria called superbug. And fun fact, did you know, before 1936, 30% of all death in the world was caused by bacteria, which is just not good. Bacteria are a menace to the medical world for a few reasons. They're difficult to find cures for because they just keep reproducing, and they can alter their DNA by pure chance. Here's the good news though, there are a few ways to cure bacterial infections. One way is to inhibit the growth of the infection or by directly killing the bacteria themselves. Scientists can do so by attacking the bacterial cell wall, or interfering with bacterial reproduction, or blocking protein production in bacteria, or even RNA synthesis. Without the use of the cell wall, bacteria cannot keep out unwanted visitors such as phages and destructive chemicals. Bacteriophages are a natural form of virus that are specific, very specific, to one species of bacteria. They attach to the outside of the bacteria and insert their viral genome into the bacteria's genome. The bacteriophage's genome effectively inhibits bacteria from replicating, producing toxins and functioning only to produce more of itself. To prevent bacteria remembering their DNA, which I talked about in CRISPR arrays in the beginning, scientists can select certain bases and certain nucleotides of the phage's DNA to alter that won't affect the phage's creation. The therapy is very selective in the bacteria it attacks and helps to effectively reduce infection and spread and side effects. The treatment is very successful compared to antibiotics and has a very low rate of the bacteria developing any form of resistance. Phages are extremely specific to one species of bacteria and do not attack healthy bacteria and healthy human cells. Phages, shown above, insert their DNA into the bacterium, and then when their DNA is copied, it is made into different parts of the bacteriophages, which are then constructed and then released, destroying the bacteria in the process. This leads to them becoming a promising method to reducing bacterial resistance and possibly eradicating harmful bacteria and their resistant genes. Plus, they look like the Apollo space capsule, which is pretty cool. Bacteria are one of the world's most primitive organisms, if you didn't already know. They are single-celled microscopic animals that thrive in incredibly diverse environments, such as the Sahara Desert, the depths of the Marianas Trench, and inside other organisms. Bacteria are one of the supporting columns of all life, because they assist in other organisms' functions and survival. The average human carries about 30 trillion human cells, which is like a lot, and carries about 100 trillion bacterial cells, which is like a lot. In terms of DNA, there are approximately 30,000 human genes, a considerable amount, and 100 times more bacterial genes, which is like a very considerable amount. Keep in mind that a single bacteria contains only like a few genes, and capable of its characteristics and functions. Bacteria play a role in every aspect of your life as they help you to digest food and eat parasitic organisms off of your skin, providing us with nutrients and many other things crucial for our survival. As bacteria can be helpful, they can also be extremely harmful to humans and other organisms. But how are microscopic organisms so small, capable of taking down larger organisms such as a human or an elephant? Bacteria have their own language called Quorum Sensing. This language allows them to count how many other bacteria of the same species there are by using special chemical molecules like hormones. These chemicals are produced by the bacteria and are received by other bacteria with the receptor on their surface specific to that one hormone chemical. Bacteria use this tool to count their numbers of a species before launching an attack on its host. Or they can use it to determine if they need to reproduce or take up a certain task in its host in a beneficial way. And they can use it to figure out if they need to break down protein in that digestive system or other helpful traits. This form of quorum sensing can be referred to as intraspecies communication, so within a species communicating. But not only can bacteria communicate within their own species, but they can also talk to other species of bacteria. All bacteria have a broad spectrum chemical and receptor that allows this to happen. Bacteria can communicate through this all-inclusive language to know how many other bacteria there are of specific species, to understand what it can work to do in the digestive tract of organisms. This form of quorum sensing can be referred to as interspecies communication. So, if bacteria can talk to each other within its subspecies or within all bacteria, and this communication allows bacteria to know when it should release toxins, doesn't that mean that we could either inhibit the production of these communicative chemicals or just block their receptor? This new idea can not only help to stop a specific species of bacterial infection, but it could also be used as a broad-spectrum form of antibiotic. Scientists are currently looking into bioactive compounds used as quorum sensing inhibitors and reducers of bacterial gene expression. Bioactive compounds such as itaconamides, lycopene, and indole are chemicals found in small amounts in plants and certain foods, such as nuts and vegetables. That could be used to block quorum sensing. So when your parents tell you to eat your veggies, I definitely suggest you listen. Biotechnology is an extremely useful set of tools because of its potential for curing infectious pathogens, viral and bacterial, types of cancer, and genetic disorders. Biotechnology today is being used to genetically edit cells in order to prevent disorders. Editing technologies are also equipping immune cells to better handle cancerous cells and viruses. Although these therapies and treatments come with good intentions, they also raise numerous ethical questions towards the morality of enhancing the human species beyond evolution. As genetic editing and therapeutic technologies improve patient survival and overall health, these techniques bring with them moral concerns towards the fact that we are playing with evolution itself, and mistakes could cause generations of trauma or create a new species of pathogens, which is just not ideal. Because of our understanding of genes, we are coming closer and closer towards the elimination of diseases and disorders, though we are still just scratching the surface of all science. Biotechnology has shown that understanding the function of genes is the key to eliminating diseases, whether they be genetic, viral, or bacterial. This is because genes can help us enhance cells to better attack pathogens. Therapies can edit mutated cells and genes, and viral vectors carrying genetic material can help inhibit bacterial reproduction and function. In the future, we can look forward to genetically tailored treatments based on an understanding of our genetic makeup, lifestyle, and environment. As genes define how even disorders and cancer cells fluctuate in growth and spread, tailored treatments can take into account genetic variability, and they can better personalize treatments. An understanding of our immune system can even influence how we are enhanced genetically to assist in human immune system protection. As we seek to eliminate more and more threats towards human survival, it has become clear that biotechnology used in clinical medicine and immune system therapies has promising potential for the future of medicine. Firstly, I'd like to thank my colleague, Pam Matthews, for dedicating hours and hours and hours over the months to reading my lecture and fixing the stupidest of grammar mistakes, and spending hours with me going over my paper and helping me organize a mass of information. Every time I worked on my paper, I felt like I'd get more and more lost and just confused and unorganized, but she really helped me to organize my thoughts and put them down on paper. Secondly, I'd like to thank Jen Henderson, of course, for keeping me organized and up to date on the deadlines. She also proved very helpful when editing my paper, giving me helpful feedback, and she was also very supportive. I'd really like to thank my external advisor, William Black, for challenging me beyond my comfort zone and exposing me to the world of science, basically helping me start my scientific research and studies at the beginning of grade 8 for science fair. You've really gone above and beyond to help me complete this lecture and teach me the ropes of scientific writing and writing a scientific paper. Not only that, but you've also spent hours helping me fix my mistakes and showing me to edit and properly speak. You've really been an awesome mentor to me. Of course, I'd like to thank my family for supporting my scientific goals and providing me with lots of food for my writing, like lots of food. You helped immensely when it came to researching, editing, and helping me write, and just helping me understand and just read stuff that a first grader could understand. They helped to spark an endless curiosity, followed by my endless stream of questions. And I'd finally like to thank Dr. Hugh Black and Dr. John Hall for your inspiration and helpful feedback. You guys have been such a source of knowledge since I was, for a long time. And finally, I'd like to thank all of you guys for listening through this boring lecture.
Very nice, very nice. I am so impressed. You pull off a really great teacher. That was not boring. But I'm not sure about the glasses, why you think that's funny. First questions are going to be from your advisors. Your first question is from Bill. We talked about a lot of things through your paper that weren't covered in your presentation. So one of the things we talked about was recent issues of COVID-19 and talking about vaccines. So you talked a little bit about vaccines. We did more in your paper really about it. But his question is, how long will it be realistically before we have a vaccine? And why do you think it's so difficult?
There's a short answer and a long answer. The short answer is between 12 to 18 months. And the reason why is because there's so many phases involved with making a vaccine. One, they actually need to make the vaccine, which can take a while. And then after that, they need to test patients, animals first, and then patients, and that can take a few months in itself, each phase. They usually start with 20 to 50 people, and then they move on to a couple hundred and then a couple thousand to test it and test its safety and effectiveness and how it affects different people, and to make sure there's no hidden variables and make sure that it doesn't come back and just even worse.
It's pretty exciting that the scientific community is sort of gathered together and sharing so much information about this to kind of try to fight it together, right? Instead of competing against each other to find it first. Okay, this question is from Hugh. I have 2 questions. He sent about 15, so we're doing our best to narrow it down. He was asking, what do you think are the most deadly diseases that the world has faced in the last 50 years?
There's been a lot. One that comes to mind first is Ebola. That was a big one that hit a while ago. I didn't go too much into diseases, like all the different diseases that we've had through history, but there's a lot out there, like different strains, like SARS. We had different types of SARS, and there's also many viruses that people don't really talk about that harmed animals and that had massive effects on animals, like mad cow disease and all these different diseases. And they may not seem very, not talked about a lot, but they still had a massive effect on humanity.
And his other question is, what are the sources of these diseases? So how do you think these viruses, how does a virus just start?
There's actually many ways. It could start as a harmless virus, and then just by pure chance or somehow its DNA mutates, and then it can now attack certain people or animals or organisms. And then it starts attacking those organisms, and it can possibly mutate further. And then it's just not very pleasant. That's what first comes to mind.
So are there natural viruses that don't harm people?
There's many viruses that harm only humans and no animals, and then vice versa. I think there's lots of viruses that we don't need to really worry about unless they somehow mutate. There's probably thousands upon thousands of different viruses. Of course, because there's thousands, I don't really know all of them.
Actually, that's not so
yeah, virology.
Okay, we're going to move to questions from me. I had a little snicker because I think at the beginning of your lecture, Scott actually thought that you were playing it up and talking gobbledygook. Like at a point there, he kind of looked around like, is this guy for real? Is he talking this? And I can tell you that you are for real. You do know your stuff. We spent hours and hours pouring through every sentence of your work, making sure that we understood it. And I've been so challenged by your work because I had to do a lot of my own research. But of all the biotechnology techniques that you researched over the year, each one had a drawback, right? So each one you'd talk about how great it was and then say, oh, and there's these drawbacks. And so much difficulty getting past the first stages with these. You called them speed bumps in your work. Which do you think is the most promising or the most likely to be implemented?
Some can be used, some are more effective against other certain diseases, but I definitely see CRISPR as being a big one. Though it's got many ethical questions, it's a lot more accurate, especially way more accurate than gene therapy, as gene therapy relies on viral vectors. CRISPR is very accurate and efficient, so I see CRISPR being a big one. And they just keep developing new methods of CRISPR. Like recently they developed CRISPR-Cas9 prime, and now that's a big new block of CRISPR that's really effective. So I think we're going to just see more and more different types, and it's just going to keep getting better.
Yes, Hunter, you're talking about antibiotic resistance. 3 things that come to mind. So we're running out of antibiotics. We know that we're running out of that. Quorum Sensing is a relatively new thing. But phage therapy has been around for actually quite a long time, but it's not something that we ever really hear about. And why isn't that more prevalent? Like, why aren't they using that?
So big drug companies, when they develop new drugs, they make a crap ton of money off of it because they can develop a patent on it. You can't really develop a patent on something that's natural. Like, it's like me developing a patent on J, which I don't really know what I use that for, but hey. but they can't make a lot of money off of that, and they're kind of all for the money because all of these developing these new therapies and treatments, it's really pricey. It's got a big price tag, so they kind of want something from it so that they can take that money and use it elsewhere, or else it's just going to be like using money somewhere and they're not really going to be getting it back. But I do see that happening in some places that don't have a lot, like Georgia. I know that's a big, they're using phages a lot in Georgia, so that's a big
I was wondering if you could tell me about the process of finding new bacteria and how is that different from finding new bacteriophage?
So to find new bacteria or use different bacteria and collect them, first they gotta go like bacteria hunting, and to do that, they can use something called like an eye chip. It's kind of like bacteria bait, like if you're trying to catch like a fish or like a worm online. But it's kind of like bacteria got something to munch on, so they're like, this is cool, what is this? So they're just like attracted to this medium, and the medium, it can change depending on like what you're searching for, I guess. But they can use an eye chip and they basically just stick it in the dirt, just in places they think will be the dirtiest, I guess, because that's when you're going to find interesting bacteria. And I guess they can just start culturing it. They can put in petri dishes, and then they just start making more. And then I guess they can just start deciding what they can use bacteria for. And it'd actually be an interesting thing to look at, just genetically editing bacteria for antibiotics. That could be something interesting. What was the second part of your question? Did I answer?
How is it different from finding new bacteriophage?
Phages, they basically just look in really dirty places like sewers and places where phages would be living. Kind of like, it's like the harsher environment, so like sewers and like places you don't really want to visit.
Dr. Thompson, I feel like I was back in university and stumbled into the wrong lecture. I was too interested to leave, but a lot of it went over my head. But a question came to mind. It seems, from what I heard, that in the near future, there's a good chance that a lot of diseases will be able to be cured by CRISPR and other ways that you were talking about. Which brings up, as you mentioned, lots of ethical questions. Who do you feel should make those decisions?
Over whether we should be using.
Yeah, when to use it and who gets it.
I see a big disagreement happening between people who think we should be using it and people who don't. I think that could cause major political issues and problems. Like, this is some big stuff, and it's not as simple as, it's not super simple. So I think it might just be like prime ministers and presidents of specific countries that either enable it or not, or it might even be within states or provincials or provinces that decide. Or it might even just be people themselves can decide if they want to use it or not, and it might not everyone is going to walk away happy from this, but there's not a lot that you can do to really monitor it.
Do you see it as inevitable that it's going to be used?
I see it as inevitable that it's going to be coming up, and yeah, that it's going to be used, but I see major conflict occurring because of it, which raises numerous ethical questions.